Okay, so we're going to go over what a COT curve is, how we're able to produce the data for a COT curve, and if we have time, we're going to move on to tandemly repeated DNA. So let's just get into the idea of what a COT curve is. So when we look at a genome, some organisms have much larger genomes than others, like humans and other large eukaryotes, like other animals. They have a lot of repetitive DNA in their genome, where if we look at smaller organisms like E. coli or yeast, they have very small, very compact genomes with very little repetition. Because when you think about it, if you're a little yeast or E. coli, pointless repetitive elements that don't help your survival, they're going to be naturally selected a lot more efficiently because they produce so much more offspring that even a small change to fitness of these organisms is going to have large ramifications for the species. And deleterious mutations like pointless repetition, which is costing the microorganisms more energy, they're going to be selected out. So essentially, genome size does not correlate to organismal complexity, especially when you think about how if we look at a couple of plants that are essentially identical, some of them can have like entire genome duplication. So some plants may be triploid where others are diploid, but we look at them, they look exactly the same. So it's an incorrect assumption to say that a larger organism will need more DNA. The C-value paradox questions why organisms that appear very similar, like those plants and flowers I just talked about, have widely different DNA contents. The majority of eukaryotic DNA is repetitive, non-coding junk DNA. And when I say most of it, I mean probably about over 70%. Like very little of our DNA actually codes for anything. I think it's only about 3% actually codes for functional proteins. The COT curve that we're going to talk about how we make it in a moment measures how much of an organism's genome is repetitive by measuring time taken for a single strand of DNA, so single-stranded DNA, to anneal to another single strand of DNA from the genome when the double-stranded DNA is split. So the time taken for a strand of DNA to find its complement. So if we think about like an entire genome of an organism, it's double-stranded. If we're to say heat it up to break the hydrogen bond between the double-stranded DNA, so we only have two single strands of DNA, and then we cut that DNA up with restriction enzymes to get a bunch of little pieces. We're basically measuring how long it takes for those pieces of DNA that we've cut up to find their complementary strand. So if you think about an organism where it only has like each of these small cut up fragments of the DNA, they only have one complement that they're able to anneal to. It'll take much longer for them to find their complement if DNA is less repetitive than if it is repetitive, because obviously more collisions, you have more complements, so they're more likely to find DNA that they're complementary to. So let's go over how exactly we work this out. So first, we mechanically shear DNA into fragments of approximately 1,000 base pairs using restriction enzymes. So we're taking the double-stranded DNA and cutting it up into 1,000 base pair units. We then denature the fragments by heating them to 95 degrees Celsius to break the hydrogen bonds and turning the double-stranded DNA that we've cut up into strands of single-stranded DNA. We then lower the temperature so that hybrids can form, so the DNA finds its complement. We then measure the amount of single-stranded DNA and double-stranded DNA present at unit time after the temperature has been lowered. So how do we measure this? To measure it, to measure the amounts of single-stranded DNA and double-stranded DNA present at unit time, the DNA is passed through what we call a hydroxyapatite column, which, if you've ever done DNA extraction, just think of it kind of like a spin column, which when you're extracting DNA and you're passing your sample through the spin column, it has a silica membrane which captures the double-stranded DNA that we've kind of extracted from whatever we're extracting DNA from. This works in a similar way, except the hydroxyapatite column only picks up double-stranded DNA, and single-stranded DNA will just pass through it. So the single-stranded DNA is passing through the column, and double-stranded DNA is binding to it. All we're doing is measuring the time taken for similar strands of DNA to anneal together. So since we in step 2, we denatured the fragments, so we broke the hydrogen bonds between the double-stranded DNA of the genome. So we would expect everything to be single-stranded, and then when we lower the temperature again, the single-stranded DNA that we've cut up is going to look for the DNA that it's complementary to and anneal to it. And once it anneals to it, it'll become double-stranded DNA, which, when we pass it through the column after a certain amount of time has passed, if it has found its complement, it will stick to the column, and if it has not, then it will remain as single-stranded DNA and pass through the column. So, like I write in step 7, the single-stranded DNA is the non-repetitive DNA, because it hasn't found a complement, because there's probably only one complement in the entirety of the genome that it can bind to. Whereas repetitive elements, they have like multiple complements that they can find, because that section of the DNA repeats in the genome. If the genome is highly repetitive, then little single-stranded DNA will pass through the column, and most will anneal to the hydroxyapatite column. So if we have a quick look at the Koch curve to the right, we can see the black line represents E. coli, which has very little repetitive DNA, like I talked about a moment ago, and a calf, so a baby cow, which is a large eukaryotic organism which has more repetitive DNA. And we can see, if we look at the y-axis, that's percentage of genome reassociation. And we can see that the calf is reassociating its genome faster than the E. coli, because if we look at the top of the y-axis, it's going in kind of the opposite order that you would expect. So 10% at the top, 100% at the bottom. The calf, because it has more repetitive DNA, its genome reassociates at a higher rate, because there are more complements in there, than the E. coli, which has very little repetitive DNA. So hopefully that's a decent explanation of Koch curves. If I've kind of muggled up the talking about it, I'll just read the slides, or maybe read the slides at the end, just so that you know kind of what it is. So that's all for that. Thank you.